So in this video, we're going to look at the different experiments that led to the understanding that DNA is the molecule of inheritance. So if you're in my class, we're making 8 boxes. We'll only fill in about 7 of these for the different experiments. So in the 1850s and 60s, Gregor Mendel was working with pea plants, and he figured out that something was being inherited from parents to offspring to pass on genetic information. But what was that thing or that factor that was actually being passed from parents to offspring? So in 1908, T.H. Morgan and a few other gentlemen scientists were working with fruit flies in New York, and they would breed flies. They would try and induce mutations with heat and chemicals and try and follow the phenotypes of the flies throughout generations. And basically what T.H. Morgan concluded in his work is that traits are inherited on chromosomes. Chromosomes in these flies were pretty easy to follow because they only have 4 chromosomes. So it's less than like humans who have 46 chromosomes. And so T.H. Morgan figured out or discovered that genes are inherited on chromosomes. He also discovered that there are things called sex-linked genes that we'll discuss more in our genetics chapter. He was also able to actually link, create a linkage map. So on the 4 chromosomes of these fruit flies, he was able to figure out where the location, basically, of each trait on the chromosomes. So pretty amazing work for scientists 100 years ago without the modern technology we have today. So if you want to take a note of T.H. Morgan in your first box, that'd be great. Now, but here's the deal. Chromosomes, that doesn't directly tell us which molecule is a molecule of inheritance, because our chromosomes are made out of both DNA and proteins. So our DNA, we have linear chromosomes as eukaryotes, and our chromosomes are wrapped around histone proteins that allow our DNA to coil up to become very condensed. So the fact that chromosomes are made of both protein and DNA did narrow down the different molecules that could be the molecule of inheritance being passed from parents to offspring, but which one is it, right? So when we look at one scientist who wasn't directly working with answering this question about inheritance, but following the Spanish flu of the 1917, 1918 or so, when a lot of the world died of influenza, we also had other infectious diseases like pneumonia that were very toxic and deadly without antibiotics. So Frederick Griffith was working on a cure for pneumonia, and he was working with, mice. And there's 2 strains of pneumonia bacteria. There's the non-pathogenic, or in this case we'll call it harmless, and there's a pathogenic strain. So when he injected the bacteria that were harmless into mice, the mice survived. However, when he took the harmful pathogenic bacteria, you can see this yellow circle around it. That's going to represent a capsule, like a sticky polysaccharide. A polysaccharide is a carbohydrate. So the pathogenic strains of pneumonia have a capsule surrounding their outside part of themselves. They're a single-celled organism, and that basically allows them to escape the immune system of the host. So it allows them to exist long enough inside of the organism to cause disease. So when he gave the mice pathogenic strains of pneumonia bacteria that have that capsule, the mice die. They become infected and die of pneumonia. So he took that he took this harmful strain of bacteria, the pathogenic pneumonia bacteria, and he killed it with heat. So you have dead bacteria, and when he injected it into the mice survived. But here's the trippy part with Griffiths' experiment. He took the non-pathogenic strain, the harmless one that did not have a capsule, and the ones that were killed by heat, that were pathogenic. And he mixed these 2 bacteria together, and what he found was when he mixed them, that the mice became infected and died. So here's the question. Griffith was like, what the heck? Something is being passed from this heat-killed bacteria. Let me go ahead and draw on this. So Griffith's question really is, there must be something passed from the pathogenic strain, even though it's dead, being passed to the harmless strain, causing it to transform and become pathogenic. So we call Griffiths' experiment the transformation experiment. So here, there's something is a transforming factor that transformed harmless bacteria to become infectious bacteria. But he didn't know what that thing was that changed the phenotype to become pathogenic. So Griffith is known for his transformation experiment. If you want to summarize that in box 2. So now we know transformation is when a bacteria that's dead can release pieces of its DNA into the environment, and then bacteria nearby can incorporate or take up some of that DNA, and now their genotype is different. But of course we didn't know this 100 years ago, 90 years ago. So now we have Avery, McCarty, and MacLeod. Fast forward to 1944, and their big question was, well, what is the transforming factor? So with Avery, McCarty, and MacLeod, their hypothesis was there must be something being passed from, so like if you have the non-pathogenic bacteria, we'll just make it a happy face, and the pathogenic bacteria that's been killed by heat, he's basically, or they are basically hypothesizing that whatever is being passed is altering the genotype. So it's passing some form of genetic information, and that new genetic information that this one is acquiring is changing its phenotype, its physical expression of the traits. So they hypothesize that there must be some kind of genetic material, genetic information changing the genotype and therefore the phenotype. But what is that genetic information? So they use some enzymes as tools. This is from the case studying class, sorry. So now here's what they did, is they took some heat-killed smooth bacteria. So this is the dead bacteria, and they treated them with different enzymes. So you have an enzyme RNase, protease, and DNase. And what they did, so let's say that this is the non-pathogenic bacteria. So when they treated the heat-killed bacteria with RNase, that's going to destroy the RNA in the heat-killed bacteria, because they're testing their hypothesis. Well, maybe RNA is a genetic material. So when they do that, though, the mice still dies. So something was still passed to the non-pathogenic strain, transforming it to be pathogenic. But you know what? They had destroyed all of the RNA, so it wasn't RNA that was being passed. Then, when we really look at the chromosomes being both protein and DNA, really those are the two that the scientists are leaning towards as legit possibilities of genetic material. And so when they take the heat-killed bacteria and they introduce protease enzymes, so protease enzymes are going to hydrolyze and break down proteins. So if protein was the genetic material, then there should be nothing transferred to the harmless bacteria. But in actuality, the mice still die. So by the mice still dying, that shows, wow, protein has been destroyed, and yet the harmless bacteria is still becoming pathogenic. So it must not be protein. Well, let's see about DNA. So then when they take the enzyme DNase, sorry, I'm trying to erase my slide. So when they take the enzyme DNase, that with your double helix would hydrolyze it and destroy the DNA, when they introduce that enzyme to the heat-killed bacteria, what they find is there is nothing transferred to the pathogenic. Nothing transferred to the harmless bacteria. They stay harmless because here you can see that the mouse lives. So through basically the process of elimination, Avery, McCarty, and MacLeod were able to confirm that DNA is the transforming factor that Griffith had seen 16 years before. Okay. So if you want to take a minute and summarize in your box 3 about Avery, McCarty, and MacLeod. Okay, and then we have Hershey and Chase here. So Hershey and Chase worked with bacteriophage viruses to basically confirm that DNA is the molecule of inheritance. So here you have some viruses. Now, this is a special virus that infects bacteria. So this type of bacteriophage virus would not infect humans. And these viruses, because viruses are made out of proteins and DNA, here they have protein coats and they have DNA. Now Hershey and Chase were curious which part of the virus actually infects the bacteria, but more realistically, like which part of the virus, is it the protein, is it the DNA that's going to carry on that genetic information for a new generation of viruses? So how can they follow? How will they know if it's DNA that infects or if it's protein that infects the bacteria? So what they do is they grow these bacteriophage viruses with their protein coats having radioactive sulfur. Because I don't know if you remember, but in protein structure, some amino acids can form disulfide bridges. So there's sulfur in some amino acids, and that's unique to proteins. DNA does not have any sulfur in it. And then the phosphorus is unique to DNA. If you remember, protein has C, H, O, and N. The nitrogen is found in the amine group, whereas DNA has C, H, O, N, and P. The phosphorus is found in the phosphate groups of a nucleotide. So they grow some, they make the DNA radioactive with phosphorus. So now they'll be able to follow these radioactive elements, either sulfur or protein, to know which one actually enters the bacterium. So if they find radioactive phosphorus inside, they'll know it's DNA infecting and carrying on the genetic information. If they find radioactive sulfur on inside, then they'll know, oh, it's sulfur. It's the protein that is carrying on the genetic information. So what they do is they allow the viruses to infect, and then the bacteria, and then they blend it. Because what they find at the bottom of the blender, you're going to have the most dense part of the solution, and that's going to be the bacteria. So by spinning it in a blender, that allows like the heavier bacteria to settle to the bottom. So when they look at their results, they find that the radioactive sulfur particles, the protein coats, are floating at the top of the solution. They did not enter into the bacteria. And what they find in the bottom of the blender, inside of the bacteria, are radioactive phosphorus molecules. So now, by seeing that there's radioactive phosphorus within the bacteria, implies or confirms that DNA is what's carrying that genetic information and passing on or carrying the info for the new generation of bacteriophage viruses. So basically, Hershey and Chase were able to confirm that DNA is the molecule of inheritance. Okay, so, if you want to go ahead and summarize that in your box 4, that is great. Now, Erwin Chargaff, he took samples of DNA from a whole bunch of different sources, from bacteria to fungus, plants, animals. And what he found with these nitrogen bases, the A, T, C, and G, was that no matter which organism he took it from, like if you look here, we can figure out, because DNA is complementary, if you know one strand of DNA, you can find the other strand. But back 50 years ago, 60 years ago, we didn't know, 70 years ago, we didn't know how DNA paired. We didn't know how it was built. So what they did, Chargaff, like now we know if I say, hey, there's 9 adenines, you can calculate how many C, G, and T there are. But this is all thanks to Chargaff, because Chargaff, all of his DNA samples, he was able to see that when you have the amount of A or adenine equals the amount of cytosine. Oh my gosh. The amount of adenine equals the amount of thymine. A goes with T and C goes with G. So no matter the source of his DNA samples, you can look here at the data. Didn't matter the species, A equaled T and C equaled G. And that was a huge step to understanding the molecular structure of DNA. So there's some sample problems for you. Okay, so if you want to summarize that in box 5. And then we have, last but not least, the structure of DNA. We have Watson and Crick and Rosalind Franklin. So Rosalind Franklin was working on X-ray crystallography of DNA, where she took this famous photo of the double helix, looking down at the double helix. This would be an X-rays bouncing off of it and leaves an imprint. And Watson and Crick had access to this photo, and they used that photo to help base their model of DNA on that information. And so Watson and Crick get the credit for discovering the structure of DNA by basing their, or by using Rosalind's photo.